its scope classification. As you can see, we have divided our scope classification according to each of the domains of this HSP90. So let's start with the N-terminal domain, but all of them will share the same class, which is an alpha and beta proteins. From the N-terminal domain, I would like to remark its ATPase domain of HSP90 chaperone. This is because the N-terminal domain, as I will be explaining later on, it's the one that acts as the ATP binding site and hydrolysis. And for the middle domain, the fold and superfamily changes, and it becomes the ribosomal S5 domain, too alike. And for the C-terminal domain, there's no additional information. So let's start with the N-terminal domain, which is an alpha-beta sandwich, as you can see in here, formed by 8 stranded beta sheets and flanked by 8 alpha helices. And you can see in here the topological diagram. So the N-terminal domain, as I have been saying, is the domain that mainly takes part on the ATP binding and hydrolysis, but it will also be important for binding some client proteins and some co-chaperones. In the closed state, the N-terminal domain will dimerize, but I will leave this for Ainhoa to explain it later on. So let's jump to the middle domain. You will see in here the representation of this domain. It's divided in 3 more subdomains, which are 2 alpha-beta-alpha sandwiches connected by the spiral of 3 helices. So this middle domain, we will analyze only its sandwiches, and you can see in here that clearly there are 5 beta strands, and in here you cannot see it more that clear, but you will have to believe me, there are 5 strands. And this was quite an issue because in literature what we found is that authors only describe 4 strands, and the last strand was really important for the change of conformations. So we decided to take a look at this conformation, and we finally concluded that our last strand was the same last strand that they were describing, so that's why we named it prima in here and in here to be faithful to what was written until the moment. So the middle domain would be like the major area for client binding, and it has a very important arginine that I would like you to take note about, because I will be explaining more and more about this arginine later on. So let's go to the C-terminal domain. This HSP90 works as a dimer, creating this 4 helix bundle in the bottom part, in the C-terminal domain. And when you look at the monomer of the C-terminal domain, you will see that it's a unique mixed alpha-beta fold. So the C-terminal domain is the main responsible for dimerization to happen. And finally, we have these domain linkers. And now you may say, well, this is not really important, just 2 small loops. Well, actually, it's important, because if you loop at them, this first loop connecting the N-terminal domain to the middle domain is only composed of 7 residues, which means that these 2 domains are not free to move indefinitely. But when you look at the other linker, it's a more long linker and it's highly mobile, which makes the protein able to adapt to different client proteins and therefore move to the different conformations of the cycle of this protein. And now that you know a bit of the overall of the structure, let's go further and let's look at the relevant regions. And we will start again with the N-terminal domain. I want you to look at this pink area in here, which is a lead, this lead-like element that is complete, which is adjacent to the ATP binding site and parallel to the beta sheet. It's not a really conserved sequence region itself, but there is a really conserved motif that you can read in here, but Pedro will be explaining much better later on. And, well, which is the importance of this region. As I have been saying, this lead is really connected to the ATP binding site. And I've been also saying that this chaperone works when binding to ATP in the N-terminal domain. So basically, this lead and this ATP binding site, what they are doing is connecting connecting the ATP binding and the ATP hydrolysis to the client binding and client expulsion of this conformation. So let's go to the client binding. First, I would like you to take a look at this 2D representation because later on I will be showing a 3D representation. But basically, the residues that are taking part in this ATP binding are glutamine, arginine, glutamate, and phenylalanine, being these last two the ones involved in the binding cleft of the ATP. And there's also this arginine that I was talking about in the middle domain, which you cannot see in here, because in here you can only see the representation of the hydrogen bonds that are being created when looking at the N-terminal domain. And this ATP-mediated binding and hydrolysis is changing the conformation of our protein, switching between pre-existing equilibrium of states and choosing the one that is more appropriate depending on the time and on how the client protein is at the moment. So now let's look at the 3D representation. You can see in here this methionine, which is doing a PP interaction, which we have measured, in fact, and it's approximately 5.3 angstroms distance. And I would also like you to know that this PDB file didn't have hydrogens, and when we tried to put them on Chimera, the program didn't know where to put them. So whenever you see these hydrogen bond distances, make sure to have into account that 1 angstrom approximately is the distance to the hydrogen bond, to the hydrogen, I'm sorry. And in here you can also see all the other hydrogen bonds and the coordination with the magnesium, but you will not be able to see much well. In fact, even Chimera didn't do these hydrogen bonds with the backbones of these residues, so we made them ourselves by analyzing the distances, and you can see in here all the hydrogen bonds that we couldn't see in the other image. And now, having seen this, we can go to the ATP hydrolysis. Okay, so the ATP hydrolysis will be, there will be 2 residues which will be very important, which are this arginine and this glutamine. So first of all, what will happen is that the arginine and the glutamine will create a hydrogen bond, and later this glutamine will be charged negatively, so it will nucleophilically attack a water molecule. This water molecule, therefore, will be charged negatively and will nucleophilically attack the phosphorus of the gamma phosphate group. So the covalent bond between the gamma phosphate group and the beta phosphate group will be broken, and we will have our products, which will be hydrogen phosphate and ADP. And these will take part on the conformational change. But not only that, we will have our arginine taking place in here, which was creating a salt bridge with the gamma phosphate group 2, and when the interaction that I've explained in a few minutes ago was broken, this arginine will be released, and therefore the conformation will change. So now let's go to the C-terminal domain. As I've been saying, this domain acts as a constitutive, well, makes the chaperone to act as a constitutive dimer. And all these residues that you have in orange are the hydrophobic ones, which are creating hydrophobic interactions, and they create like a kind of ceiling, hydrophobic ceiling in here that will be surrounded on top by these hydrophilic residues that will be in contact with the surface. And also I would like to remark this hydrogen bond happening between glutamine and asparagine in here. And if you switch the molecule, you will see exactly the same interactions. Maybe in here you can see much better the hydrophobicity marked in red. And what I would also like to remark about this C-terminal domain is a conserved motif in here, which has nothing to do with this dimerization. It's a highly conserved motif, which has been thought to take some action when binding to cochaperones. And finally, this H21 helix, which I would also like to remember. I would also like you to remember because in a few slides I'm going to be talking about it. So it's just basically a conserved hydrophobic residue, not in sequence, but in hydrophobicity. And this is the central cleft. So the central cleft would be like the part of this chaperone where the client protein will be bound. And it's created by certain domains of each domain of the structure. So we will have the N-terminal domain creating this hydrophobic cleft, which will be formed by these amino acids clustering with these other amino acids. In the in this middle domain, we will have this hydrophobic region over here, which is called SRC loop. And finally, this in the C-terminal domain, we'll have H21, which is the one that I was asking you to remember. And all these will conformate the central cleft for the client protein to be bound. So actually, this hydrophobicity is really conserved in structure, but it's not that well conserved in sequence. And what this may mean is that client proteins can be bound, but what is important is the hydrophobicity and not the residues itself. And now I'm gonna leave Pedro to explain this.
Thank you, Mariana.